So remember we were talking about red blood cells, and so we want to keep red blood cells as close to ideal as possible, because if you don't keep it close at ideal, you wind up with problems. We don't want too many because the blood gets too thick. You get this thick viscosity, and you should remember that if viscosity goes up, then flow goes down. And if you increase the viscosity, you're also going to have to increase the pressure to push the blood around. So you're going to make the heart work harder. You're going to increase blood pressure. And you're going to be at more of a risk for things like heart attack and strokes. And so we would not want too many red blood cells. We also don't want too few red blood cells, because if too few red blood cells is going to be too little oxygen being delivered. And when you have too little oxygen, that means that all of the tissues are going to suffer. They're going to be hypoxic. In other words, they're going to have low oxygen. And without oxygen, remember, you can't make ATP. And so ATP levels are going to go down. Without ATP, you can't really do anything, because that's the energy source of the cell. And so we want to keep red blood cell number as close to ideal as possible. But red blood cells only live or circulate about 120 days. Remember, they don't have a nucleus, they don't have any organelles, they can't multiply, they can't fix themselves. And so after 120 days, they become very, very fragile and they break down. And so if you're going to have them breaking down, you also have to make them. And so the organ that's in charge of regulating how many red blood cells we have is the kidneys. And the kidneys make a hormone. So even though this was not one of our endocrine organs when we talked about endocrine organs, this one is. It's an endocrine organ. And the hormone that it makes is called EPO. EPO stands for erythropoietin. And erythropoietin, or EPO, is water soluble, so we should remember how it works and all that. Nothing is any different. And so what happens is when the kidneys come into contact with low oxygen levels, well, what that means is low oxygen. It means that there's not enough red blood cells. There's too few red blood cells to carry the amount of oxygen that you need. And when it gets to the kidneys, what happens is those kidneys detect the reduced oxygen levels and they release that hormone, EPO. Again, EPO stands for erythropoietin. But anyway, that EPO, or erythropoietin, goes to the red bone marrow. And there, what it does is it stimulates the red bone marrow to make more red blood cells. And so remember, there's a stem cell, and if we give it EPO, what's going to happen is it's going to go through that whole process, and we're going to wind up with more red blood cells. And if you have more red blood cells, you're going to have more oxygen carrying capacity. And so oxygen levels are going to go back up to where they were. And as soon as that happens, then the kidney is going to stop releasing EPO. And so we're going to slow down red blood cell production until what happens is it goes down again, release EPO again, and so on. And so if you look at it, and this is just a different way of looking at it, but here's the low oxygen. We're going to release this EPO, so EPO is going to go up. Again, EPO is going to go to the red bone marrow. Red bone marrow is going to increase the number of red blood cells, and they're going to wind up in circulation. And they're going to circulate for about 120 days. We talked about that all last time. Any questions about any of that? That's not new material. What we didn't talk about last time, and somehow I skipped, is what happens to them after 120 days. What's their fate? Remember, they're going to be gotten rid of, and so they're going to be destroyed in some way or another. Well, what happens is, as they get older, that spectrin in their, that's a protein in their membrane, is what allows them to flex and bend and stack up and all that. Spectrin begins to wear out. And as it wears out, remember, they can't fix themselves. What's going to happen is the cells are going to become rigid and fragile. And so what's going to happen to the membrane is more likely to break down. Not only that, but inside the red blood cells, hemoglobin begins to degenerate. Remember, it's also a protein. And proteins don't last forever, but these cells cannot make anything new. And so that protein is going to begin to break down inside the red blood cell. Because remember what a red blood cell is basically just a little sac of hemoglobin. It's just a little sac full of hemoglobin. Remember, there's about 280 million molecules of hemoglobin in just one red blood cell. And so what's going to happen is they're going to begin to break down. Well, remember when we talked about capillaries? We talked about capillaries, and we said capillaries were tiny, tiny little blood vessels, and they're about 8 microns in diameter. And I even said, think about this, that red blood cells are 8 microns in diameter, and so they have to go through capillaries one at a time. They go through single file. But there are some capillaries in the body, mostly in the spleen, but then in a few other places too, the bone marrow and the liver. These capillaries are smaller, so they're not 8 microns. They're only about 3 to 5 microns in diameter. Well, think what's going to happen. Remember, our red blood cell can flex and bend, and it's going to go through like this. It's going to get squished and go through almost like a sausage. Well, once that red blood cell becomes rigid and fragile, what's going to happen is it's going to rupture. Well, which of these two capillaries, just plain old body capillaries or spleen capillaries, do you think they're more likely for the red blood cells to rupture? Which one? You think it's more likely to happen in these or in those in the spleen? Well, it's going to be the spleen because they're under much, much more pressure. You're squishing them. And so the spleen is mostly where this rupture happens. And so in the spleen, there are a ton of macrophages. Remember, these are monocytes that have turned into macrophages. They're white blood cells. There's a ton of them in here. And what those monocytes are going to do is they're going to engulf the red blood cell. So here's the old, damaged, ruptured red blood cells, even some that haven't ruptured. They're going to get engulfed by these macrophages. And then something's going to happen. When you engulf the red blood cell and you eat it, you break down the cell membrane. And so all that hemoglobin is going to be released. Well, this is very old hemoglobin, and it's not very good. And so we're not going to recycle the hemoglobin. But what we are going to do is recycle the parts of it. Remember, hemoglobin, when you look at it, it's got 4 peptide chains. And then in the middle of it is that heme, which has a porphyrin ring. And then in the center of that, that's where the iron is. So there's a bunch of good stuff here. So what happens first is we're going to separate the peptide chain from the porphyrin ring, from the heme. So we're going to wind up with 4 hemes, and we're going to wind up with 4 peptide chains. And what's going to happen to these 4 peptide chains is they're going to be digested. They're going to be broken down into the individual amino acids. And those amino acids are really valuable. We wouldn't want to get rid of them. So those amino acids are recycled. So that's what happens to that part of it. Let's look at the heme. Well, remember, heme is this big ring structure like crazy with lots of double bonds and so on. And in the very center of it is an iron. And we're going to have 4 of these, remember, for every hemoglobin. Well, this iron is also very valuable. And so what we're going to do is we're going to remove the iron and we're going to recycle it as well. So we're going to send it back to the bone marrow so it can be used again. It doesn't wear out, it's a metal. And so it can be used again to make more hemoglobin. So we've gotten rid of the peptide chains. We remember we turned them into amino acids and we recycled the amino acids. We've gotten rid of the iron. We're going to send it and we're going to use it again to make more. All that we're left with now is that porphyrin ring. That ring, and it's a very complicated ring. Well, that complicated ring, because it has so many double bonds and it's got nitrogen in it, it's got carbon in it, it's very difficult to break down. And another thing about it is, if it runs around loose, it's actually toxic. It can clog up vessels, it can clog up the kidneys, it can do all sorts of bad things. So this heme needs to be gotten rid of as well. The way the heme is gotten rid of is that it's converted to less and less toxic forms. So first, that ring, that porphyrin ring, is converted into biliverdin. Well, if any of you speak Spanish or some other Romantic language, you know that verde means green, and it is green. It's green in color. And some of that winds up in the bile that we secrete from our livers, but most of it is converted into an even less toxic form called bilirubin. And bilirubin is yellow in color. So biliverdin is green and bilirubin is yellow. So if you look at bile, you look at the gallbladder and you look at what comes out of it, bile, this yellow-green color. Sort of a chartreuse, if you know what that is. So that gets secreted. It gets secreted. And what it does is it gets secreted into your intestines. So it goes into your small intestine. And then as it moves through the small intestine, it moves into the large intestine. And there's tons of bacteria in the large intestine. And they're actually going to use it for something. What they're going to do is they're going to take this part off. And that becomes useful to them. They can use it as an energy source. But what happens when you take it off, you're left with something else. You're left with something called urobilinogen, and stercobilinogen. And then what's going to happen to that is urobilinogen is going to get converted into urobilin. And urobilin gets reabsorbed back into the body. And it winds up in your urine. Think about what color urine is. Unless you're sick or unless you're taking some sort of medicine or maybe a lot of vitamins or something, urine has a definite color. Well, the color of urine is caused by this, by urobilin. This other one, though, stays in the intestines and it gets converted to stercobilin. And stercobilin colors our feces. So again, if you think about normal feces, you're not sick, you haven't eaten anything weird or whatever, our feces have a particular color. And that color is caused by stercobilin. So, both the color of our urine and the color of our feces is due to the breakdown product of red blood cells, due to the breakdown product of hemoglobin. So, here's that. It's a kind of a little pathway, not very complex, but remember what happens to hemoglobin. First, we're going to take off the protein part, the globin part. And then we're going to be left with the heme. There's the heme. And then we're going to take the heme, we're going to take off the iron. And these two things here are going to be recycled. We're going to use them to make more hemoglobin or make more proteins or whatever. And then that heme, here it is. All that's left is that ring. And that ring, remember, is a little bit toxic. It also can clog up things and so on. So we need to excrete it. But before it's excreted, it's converted first into biliverdin, then bilirubin. It goes in intestines. It gets converted to urobilinogen and stercobilinogen. Urobilinogen is reabsorbed and it goes into the urine. Stercobilinogen becomes stercobilin and it goes into the feces. Any questions about it? Okay, so that's what happens to the red blood cells. Remember, they're going to live about 120 days. They get fragile. They get eaten by white blood cells. And then the hemoglobin is going to get, some parts are going to be recycled and some parts are going to be excreted. And remember, we lose about 3 million red blood cells every single second, which means that we have to make 3 million red blood cells every single second. So here's another view, same thing. So here's our old, damaged red blood cells. They're engulfed by the macrophages in places like the spleen, also the liver, and also the bone marrow, but mostly the spleen. In fact, so much of this happens in the spleen that it's nicknamed the red blood cell graveyard, because that's where most of them are going to wind up when they die. We're going to separate it. That part's going to get recycled. We turn it into amino acids. This part, we're going to pull off the iron. That's going to get recycled, and the rest is going to get converted into biliverdin and then bilirubin. It's going to get excreted. And in the feces, remember, we're going to get stercobilin and so on. Any questions? Okay, well, remember, we want to keep the number as constant as possible. If we have too many, that's bad. If we have too few, that's bad. So let's look at some disorders that are like that. Let's look at too few first. So when you have too few red blood cells, it's called anemia. And so if you have too few, that means that your oxygen carrying capacity is going to be very low. So look, if I centrifuge blood, remember this is whole blood, we already know what's going to happen. The red blood cells are going to wind up at the bottom. So if you look at the normal amount, this is how many red blood cells there are. And depending on if you're male or female, it's going to be different, but let's say it's about 40%. But if you're anemic, look what happens when you centrifuge that blood. This person only has that much of their blood as red blood cells. And to me, that looks like about a third of that. So let's say that's about 13%. Well, it should be very obvious that this many red blood cells can carry a lot more oxygen than this many red blood cells. And so this person is going to, all over their body, they're going to suffer from this low oxygen. So this person is going to be very tired. They're going to have no energy at all. They're going to feel malaise or slightly sick all the time. They're going to be pale. They're going to have shortness of breath. They might have things like chills even. But the symptoms are all about lack of oxygen. Well, there are over 100 different types of anemia. There are a bunch of them. So rather than talking about all those, because each one depends on what's going on, but it could be something like low iron. But it doesn't have to be low iron. So if someone's anemic, you can't just tell them to take iron pills and that'll fix it. It could be low vitamins. It could be that their red blood cells are just not being produced. In other words, there's something wrong with the bone marrow. It could be that they don't have enough amino acids to make the hemoglobin. There's all kinds of things that can cause anemia. So anemia is not a disease, it's a symptom. So if someone says they're anemic, it's a symptom of something else. And so to fix the anemia, you have to figure out what is the actual disease or what is the actual problem. Any questions about that? So the opposite of that is when you have too many red blood cells. So look, if here's our blood again, and there's our normal. Look, there was our anemia, but here's too many. It's called polycythemia. And so this is like, it's going to cause the blood to be very, very thick. In other words, you're going to increase the viscosity. Again, we already talked about it, but if you increase the viscosity, you're going to wind up with a decrease in flow, and you're going to wind up with an increase in blood pressure. And so these people are at high risk from things like heart attack and stroke. Now, this in itself is not a very common disease. There are people who have it, but usually the people who exhibit polycythemia do it on purpose for some reason. And a lot of athletes, especially athletes that are in endurance sports, like cycling or something like that, what they will do is over time, they'll take blood out, their own blood out of their system, and they'll store the blood. And then right before a race, they'll take that stored blood and they put it back in their body. And so what happens is all of a sudden when you put all those extra red blood cells back in the body, this is what happens. The red blood cell number shoots up. Well, why would they do that? They do it because the more red blood cells you have, the more oxygen carrying capacity you have. And so even though they're increasing their risk for things like stroke and heart attack, they've got a lot of oxygen. So during that race, they don't get as fatigued, they don't tire out as quickly, so they can go a lot further at a lot higher pace. You can also do the exact same thing by just injecting something. What would you inject? Do you know? We've already talked about it, but if I wanted to increase red blood cell number, what would I inject? What would I give myself? What increases the number of red blood cells? It's a hormone. What is it? Anybody know? It's EPO. So if I inject EPO, it's going to go to the bone marrow and it's going to speed up red blood cell production. And so now, instead of making this many red blood cells when I use with EPO, I'm going to make that many red blood cells because it's extra EPO. It's excess. Does that make sense to everyone? So if you look at those athletes are constantly being tested. And they test for EPO or EPO-like compounds. They do this quick and dirty thing, the hematocrit, to see what their normal is. It should be something like this, not something like this. Any questions? Another thing that can go wrong with red blood cells is a genetic disease. A genetic disease. In other words, you're going to get it from your parents. So if you look at this person and this person, they carry a trait. So remember, we have two chromosomes. They're in pairs. So one of their chromosomes is normal, but the other one has this abnormal gene on it. And so when these two people get together to have a baby, look, this person is normal because they got normal from both their parents. This person and this person carry the trait. But this person has two of these abnormal genes. And when you have two of these abnormal genes, you get the disease. The disease is called sickle cell anemia. Sickle cell anemia is caused by a mutation. And the mutation is a very simple mutation. It's in one amino acid, just one out of the whole proteins. So here's the normal DNA. But look, they're opposite here. They're reversed. And so just that one mutation right there changes this amino acid to this amino acid. And what it does is it makes the hemoglobin less stable. Less stable. And so what happens is if you get in a low oxygen situation, that could be due to exercise, it could be due to flying in an airplane, it could be due to living at a high altitude or whatever. But when you get in this low oxygen situation, this hemoglobin is less stable. And what it does is it unfolds. Remember, there's supposed to be 4 peptide chains, and they look like this. But what happens is they unfold. So they're like that. And when that happens, and all those hemoglobins do that inside the red blood cell changes shape. And the shape it picks up is something like this. And it looks like a sickle, so that's what it's called, sickle cell anemia. Well, think about this. This and this, they both have to go through capillaries. Remember, the capillaries are so small, they have to go through one at a time. And some of them are even smaller than that. And so if you think about it, this is going to be able to get through there because it's smooth, it's round. There's nothing to catch on things. But look at this. It has these pointy parts, and those pointy parts catch on things. And so what happens is you get lots and lots and lots of tiny little blockages all over the body. Lots and lots and lots of these things. And depending on where it is, and if there's any collateral circulation or not, these little tiny blockages can create all kinds of problems in lots of different organs. And so we can have multiple organs involved. And so when people have this happen, then they wind up with lots of pain, they wind up with lots of inflammation, they wind up with lots of little blockages so that the cells are not getting the oxygen they're supposed to. And so those cells are hypoxic. All of that happens. And so these people wind up with high fevers. They wind up with an inability to do anything. Their joints hurt like crazy. So they're in tons of pain. And the only way to fix this is by transfusions. And so these people have to have a lot of transfusions because transfusions are going to have normal red blood cells, normal hemoglobin. But a transfusion doesn't last forever because remember, the cells only live about 120 days and we're constantly getting rid of them and constantly making new ones. But the new ones that we make are going to be this type. And so people who have this issue have lifelong problems. So you might wonder, why in the world would we have this mutation? Well, there's a reason for it, and that reason has to do with malaria. So there are some places in the world where malaria is endemic. In other words, it's there and everybody's exposed to this parasite. Well, it turns out that if you're this person, you get malaria really easily and you get a very bad case of it. And a lot of these people die from malaria. Well, remember, we also got this person. This person has sickle cell disease. And so they're going to have issues their whole life, and a lot of these are going to die. But what turns out is if you're this people, you don't get sickle cell, and you also are resistant to malaria. In other words, if you get malaria, it's a lot weaker, a lot less serious case. And so if you think about the whole population, if these people die, and these people die, and these people live, this gene is going to hang around in the population because there's an advantage to it. Does that make sense to everyone? Any questions? So, which populations would you expect to see sickle cell anemia in? It's not very common in whites. Not very common at all. Because whites, their ancestors didn't come from places where malaria is. So you don't see it in whites. You see it in a lot of African Americans because there's tons of malaria in Africa. And you can also see it in South Americans and some other places like that because there's also tons of malaria in those places as well. Any questions? Does it make sense? Okay, let's look at something else about blood. And that is, we have different blood types. So a little bit of history about this. So we've known for a really, really long time, even way back, say, in the Middle Ages, that if you get hurt and you lose a lot of blood, you're probably going to die from blood loss. And people knew that from animals, and they also knew it from people. And so even back then, people knew that if I took blood out of one person and I put it in another person, it should work. And so they actually did transfusions way back when, and they took it directly out of one person and they put it into another person. So this person then is getting this person's blood. So we have a donor and we have a recipient. Well, it sounds like a good idea. And for some people, it was a good idea. But just from this transfusion, you had a 50-50 chance of dying. So some people, when you did the transfusion, this guy would get up and walk around. He was okay. But some people, when you did the transfusion, they would instantly, almost instantly, just die right there while it was happening. And nobody understood why this was. Well, one of the first people to investigate this was a doctor. And what he decided to do was he wanted to see what was going on. He was actually a doctor that was involved in yellow fever. But that has nothing to do with it. That's just where this was all at. And so he knew about this transfusion things because with yellow fever, you have to have transfusions. So what he decided to do was he decided to test blood. So what he did was he collected blood from himself and all of his patients and all of his staff. And so he had tons of these blood samples. He had them all in tubes, tons of blood vessels or blood samples. So he collected the blood. Here's the blood here, and here's the blood. And then what he did was he took blood out of one tube and he put it in another one. So in other words, he took a little bit of blood out here with a pipette, and then he would put it in there, and he would put that blood in there, and he would put that blood in there, and so on. And what he found out was about half the time, nothing happened. And so if you looked at the blood after you put it in there, it just looked like blood. Nothing happened. But the other half of the time when he put it in there, what would happen is the blood would clump up. It would clump up like crazy. So one person would be normal, and then the next one would clump up, and form these big clumps. Normal and then clump. So what he found out by mixing up all of these blood and figuring it out who when you give to this and who when you give to that, and he would do it with every single one of them. What he figured out were that there were different types of blood. He didn't know what was causing it, but he figured out there were different types of blood, and he decided to name them with letters. And so he wound up with type A, type B, type AB, and then there was one he called type 0. Well, we still use these designations even today, but 0 is somehow over time just been shortened to O. But 0 actually means a lot more than O does. What he meant by 0 is that it's lacking an A and it's lacking a B. They don't have them. There's no A and no B. So 0 actually means more than O does. But let's look at this. So here are these types. And what they're caused by, and he didn't figure this out, somebody else did, is that there are antigens, proteins on their surface. So when you look at them, they look like this. So there's a red blood cell of someone who's type A. So these are A antigens. There's someone who's type B. So these are B antigens, B proteins. But look, this person has this one. It has A, and it has this one. It also has B. But look, there's our type 0 or type O. It doesn't have either one of them. So that was a big discovery, but it still did not explain why it clumps up. Why do they clump? And that took him much later in a different person. And what he found out was, well, here's where you get your blood type from. You get it from both your parents. And so you can inherit A, B, or you can inherit the 0. But what happened was a completely different person figured out something else. And what that person figured out was that in our guts, so here's our intestine, there are bacteria in there, thousands and thousands and thousands of bacteria. And I draw a bunch, but there's millions of them in there. Well, some of these bacteria are special little bacteria because these bacteria have proteins on their surface too. And the protein on their surface is exactly like the protein on the red cell. So some of these bacteria have the protein A on them. Some of these bacteria have the protein B on them. Well, think about this. Here is the person who's type A. So we already know on their red cells, they have A. All over it, A. Well, think about this. If this is this person, to them, to this particular person, what is A? Is it self, which means it's part of them, or is it non-self? Which one is it for this person? Self or non-self? It's on their red blood cells. So is it self or non-self? Anybody know? It's self. So for this person, A is self. So look, normally when we see bacteria, we want to kill them because they can hurt us. And we make these things called antibodies to attack these bacteria. But look at this. This antibody is specific to a particular protein. So if A is self, look, that's also A. Well, when I see this, I want to make an antibody. But if this antibody were to attack that, it would also attack that. In other words, I'd be killing my own cells. So can I make an anti-A antibody? If I do, I'm going to kill my own tissues. So can I do that? Yes or no? I remember, if I make this antibody to attack this A, it's also going to kill this A. It's going to kill my red blood cell. Do I want to go around killing all my red blood cells? No. So can I make an anti-A antibody? I cannot, because A is self. But here's this person. Look at this. They have these bacteria in there too. They have B. So for this person, is B self or non-self? Which one is it? It's not present on their red cells. So is it self or non-self? It's non-self. So can I make antibodies against this? Remember, this is non-self. Can I make something to kill those bacteria? Yes. Yes, I can make anti-B antibodies. So floating around in this person's plasma is going to be anti-B antibodies. So if we look at this, look, this person is type A. And what I'm going to have on here is A, but I cannot make anti-A antibodies. But B is non-self, and so this person can have anti-B antibodies. That makes sense? Well, let's go back. What if I'm type B? So again, here's our gut over here, and here's these bacteria. And this bacteria has on it A, and this bacteria has on it B. So let's look at this now. For this person, what is B? Is it self or non-self? This person is type B. So on their red cells, they have B. So what is B? Self or non-self? That's exactly right. B is self. So can I make any anti-B antibodies? No, I cannot. But for this person, what is A? Is it self or non-self? It's non-self. And so can I make anti-A antibodies? Can I make anti-A antibodies? Yes. And that's exactly what I'm going to do. I'm going to make antibodies against this A, and they're going to be floating around in my plasma. So if you look at this person's blood, look, this person is type B. B is self, but A is not self, and so I'm going to make anti-A antibodies, and they're going to float around in the blood. But let's go back. What if this person here is type AB? So for this person, what is A? Is it self or non-self? It's self. What is B? Self or non-self? It's also self, because remember, on their red cells, they have both A and B. But they're still going to have these bacteria. But again, this bacteria has A on it. This bacteria has B on it. Well, if A is self, can I make antibodies against A? No. If B is self, can I make antibodies against B? No. So when I look in their plasma, I'm not going to have any anti-A, but I'm also not going to have any anti-B. There's not going to be either one, anti-A or anti-B. So when I look at their blood, look, here is the A itself. Here is the B itself. So I cannot make anti-A or anti-B antibodies. So neither one of those is going to be floating around in their plasma. But here's one more case. So this person is type O, or 0 is a better way of saying it, I think. Type O. So if I look at that person's red cells, look, they look like this. There's no A's and no B's. So think about this. For this person, what is A? Is it self or non-self? It's non-self. What is B for this person? Self or non-self? It's also non-self. So they still have these bacteria floating around in their gut with A and with B. So when their body sees these, am I going to make antibodies against this A? Yes. Am I going to make antibodies against this B? Yes. And so in their plasma, I'm going to have anti-A and anti-B floating around in there. So when you look at this person, this is what their blood looks like. So look, there's no A's, no B's on there. They're neither one of them. And so I can make an antibody against A, and I can make an antibody against B. So when you look at their plasma, it's going to have both floating around in there. Well, antibodies attack antigens. So if we go back over here, remember here is A, this bacteria. So if we look at A, so I'm going to make an antibody that attacks that. If it's on a red cell and that antibody is around, it's also going to attack that. So if I were to put the wrong blood type, I mix it, look what's going to happen. If I mix this, which is type A, and I put it in here, which is type B, remember type B has got anti-A antibodies in it. And if I put A in anti-A, what they do is they clump up. It looks like this. So this is type A right here, is type A. And if I put type A in a place where there's anti-A, this is what's going to happen. They're going to clump up. And so if you put the wrong blood type in the wrong person, these antibodies attack. So if I put this in here, those antibodies are going to attack that. If I put this in here, these antibodies are going to attack that. If I put this in here, look, these antibodies are going to attack these, and these antibodies are going to attack those. And this is what's going to happen. And what happens is you get what's called an acute hemolytic reaction. It's actually considered to be a hypersensitivity of the immune system. It's called a type 2 allergy. And we'll get to allergies later on. But look what happens all over this person's body. It's going to clump up all over, everywhere, in every single blood vessel. And that blood just cannot be pushed around. And so all over this person's body, we're going to get all kinds of weird symptoms. And so you're going to get things like fever and chills. Heart rate's going to go up like crazy. Blood vessels, blood pressure is going to drop. And this person basically is probably going to die unless something is done very quickly. So again, cells with this antigen A are type A. Cells with this protein antigen B are type B. Cells that have both are type AB, and cells with neither are type O. Any questions about any of this? So does everybody understand what a type A person has? On their red cells, they have A, but also because of those gut bacteria, they're going to have anti-B. Type B is going to have B on their red cells, but they're also going to have anti-A. Type AB is going to have A and B on their red cells. And since both of these are self, they cannot have either one of these. So they have neither. And then type O, there's no A and there's no B. And so they're going to have both antibodies. So we have to think about that when we think about giving someone a transfusion. So this person is type A. There's their A on their red cells, and there's the anti-B. Look, you're going to give them a transfusion. But look what you transfused them with. That's type B blood. Type B blood has B on it, which is exactly what these things attack. And so as soon as that blood gets in here, That's what's going to happen. They're going to clump up like crazy. And then we're going to wind up destroying those cells. And all that hemoglobin is going to be released. And that is what causes all these symptoms. So transfusions are really an important way to provide blood for someone who needs it. But we cannot give them the wrong blood. And so we have to know what the types are, and then we have to know which types can be given to which other types. Otherwise, this is going to happen. So in order to do that, we need to type the blood. What type of antibodies does type AB have? They have neither one. They don't have any anti-A or anti-B. If we go back to that picture, remember they have neither. Make sense? So look, I want to know what your blood type is. Well, when I look in a tube, all I see is blood. I can't see these antigens on here. They're just too small. So the way I'm going to figure out what your blood type is I'm going to use antibodies just like these. So what happens is I'm going to take something like a rabbit or something. So if you think about a rabbit, here's a rabbit, not a very good one, but here's a rabbit. What I'm going to do is I'm going to inject that rabbit with your blood. So let's say you're type A. So remember, type A people look like this right there. So I'm going to take that blood and I put it into a rabbit. What's going to happen is rabbits are not type A. They don't have that same blood types. And so the rabbit is going to make these antibodies. So they're going to be floating around in the plasma of this rabbit. What I'm going to do is I'm going to bleed this rabbit. I'm going to take its blood. So I'm going to use a syringe, draw blood, put it in a tube. And then what I'm going to do is I'm going to separate out these antibodies. And I'm going to put those antibodies in a bottle. And so there's nothing in this bottle except anti-A antibodies. Anti-A. What is the only thing these antibodies will attack? They're anti-A. What do they attack? They attack A. And so if I put this near A, remember this is going to happen. It's going to clump up. And then I'm going to do the same thing with a different rabbit. So here's my rabbit again. And this time I'm going to inject B in there. And I'm going to bleed this rabbit. And what I'm going to get out is blood. And then I'm going, remember this rabbit B is he's going to make anti-B. So when I take the blood, I'm going to separate it out. And I'm going to put those in a jar. And so all that's in here are anti-B antibodies. So what are the only thing anti-B antibodies will attack? Anti-B. What do they attack? They attack B. And so what now I've got is a way to see if there's A and a way to see if there's B. So look, I want to find out what these people's blood types are. So here's person 1, 2, 3, and 4. Well, this is just plain blood with nothing in it. So that's what blood's supposed to look like if it hasn't clumped up. So I'm going to take 1, 2, 3, 4 drops of this person's blood. I'm going to put it on a slide. So here's my slide, and I'm going to put it in this drops. And then I'm going to add to it that anti-A that I got from the rabbit. And I'm going to add to a different drop, anti-B, that I got from the rabbit. And then I'm going to look and see what happens. So look, this one clumped. It clumped up. You can see the clumps. There they are. So is A present? Remember, anti-A attacks A. Is A present? Think about it. Anti-A attacks A and makes it clump up. So yes, A is present. But look, I'm going to take that same person's blood and I'm going to mix it with anti-B, and I did not get any clumps. So, in other words, no attacking. So is B present? No, it's not present. And if I go down this whole list, we should be able to figure out. If I look at this one, this person has type A. Look, they're A. This person, look, the B clumped. When I put the anti-B in there, it clumps. So this person is type B. And if I look at this one, look, A clumped and B clumped. So what type are they? They're AB. But look at this last person. Anti-A, when I put it in there, did not cause clumping. Anti-B, when I put it in there, that did not cause clumping, which means there's no A, and that means there's no B. And what is the type that has no A and no B? It's O. Is this making sense to everyone? There's one more thing that we have not talked about. On our red cells, we also have another protein, and it's called RH. Sometimes it's now called D. But RH stands for rhesus monkey because that's the first place it was discovered. Well, people also have RH, and you either have it, which means it's on your red cells, or you don't have it, which means it's not on your red cells. So look, this person has it, this person does not. So this person is said to be RH positive, and this person is said to be RH negative. Well, RH has absolutely nothing to do with A and B. So look, this person could be this. So they could be A on here and RH on here. Or it could be A on here and no RH. Another person could be B on here and they have RH. But a different person could be B without any RH. Same thing for AB. They can be A, B, and have RH, or they can be A and B without any RH. Same thing for O. So if I look at what this person's blood type is, look, they're type A and they're RH positive. We can shorten that to just say they're A positive. So what is this person? They're still type A, but they don't have RH. So they're A, RH negative. Or we could say this person, shorten it to A negative. And that's true for all of these. Does it make sense? So we also need to worry about RH. Or like I said, sometimes it's called D. But this is RH. So here's what we're going to do. We're going to do exactly the same thing we did before. We're going to take a rabbit And we're going to inject this rabbit with RH. And then we're going to collect blood from our rabbit. And then when we take the blood out, we're going to extract anti-RH antibodies, just like we did with those other things. And we're going to put them in a bottle. And so in that bottle, all that's in there is anti-RH. The only thing anti-RH will attack is RH. And then we're going to use it to figure out, is this person RH positive or RH negative? Well, remember, if it clumps up, that means the RH is present. So what is this person, positive or negative? They're positive. What is this person? Positive. What is this person? Positive. What is this person? Negative. So when we determine a blood type, we need to put these two things together. So remember, this person was type A, and they're RH positive, so they're A positive. This person is type B, and they're RH positive, so they're B positive. This person was type AB positive, and so, AB and positive, so they're AB positive. But this person did not have any A, did not have any B, and they don't have any RH, so they're O negative. Everybody think they could read a blood type? So now that we know what a blood type is, we can figure out our patients. Here's our patient. And we can figure out the blood that was given to a blood bank, the donor's blood type. And now we can figure out who can give to whom. And so if you look at this, there's the donor, there's the patient. So if my donor is this person, look, they can give to this, to this, to this, to this, to this, to this, to this. But if they're this person, they can only give to this, 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 and this. If I'm a patient, look, this patient can receive blood from every one of these. But if I'm this patient, I can only receive blood from that. And so this is a little chart to show you who can give to whom. And so you need to be able to figure this out. And here is a little sort of clue-like thing. Negatives can give to positives, but positives cannot give to negatives. And then remember, A can give to A, B can give to B, and so on. So look, A negative can give to A negative, but A positive, look the wrong direction. A negative can give to A negative, but A positive cannot give to A negative. Negatives can give to positives, but positives cannot give to negatives. So, like I said, you need to be able to figure this out. Well, look at these blood types. There's two blood types here that are kind of special. One of them is this one, because look, it can give to every single person, no matter what their blood type is. So sometimes that is said to be the universal donor. The universal donor is O negative. And O negative blood is very, very valuable. And the reason it's so valuable is because you can give it to anybody in an emergency. So you don't have to worry about blood typing them or anything like that. You can just give it to them. And so you can stop things like your ambulance or something like that with this blood and give it. Or you can use it in an emergency room without having to wait to figure out what their blood types are. So it's a valuable blood type. And if a blood bank finds out that you're O negative, they're going to call you all the time. They're going to call and call and call and call because they need your blood. The other blood type that's special is this one. And the reason this one is special is because, look, this patient can get blood from anybody. You don't have to type it. You don't have to do it. Everybody can give this person blood. So every blood type can donate to this one. So sometimes this is said to be the universal recipient or universal acceptor. That's AB positive. And they can receive blood from anybody. Well, actually, this person is very, very lucky because there's no way they're ever going to get a transfusion reaction, even if somebody screws up in an emergency room or something. They're never going to get a transfusion reaction. They're very lucky. But this blood is also not valuable to the blood banks. Not valuable at all. Because look who this blood type can give to. They can only give, look, if you're a donor, you can only give to this blood type. So none of these other blood types can accept your blood. So this is the least valuable blood, actually, in terms of that. Any questions about blood types? Well, if you want to know who has which blood, it really depends on the population, where you came from. The most common blood type is O positive, but that changes depending on where you are and where you came from. So if you look at, I guess I don't have it on here, but if you look at Native American Indians, almost none of them have Rh. They don't have any. And so it just depends. Okay, there's one more thing I want to talk about blood types. Remember we said that if you give type A blood to someone who's type B, almost instantly you're going to get this clumping up. That's because there's already floating around in their plasma these antibodies that are going to attack it. Remember, those antibodies came or are there because we have these gut bacteria. That's the only reason we have these antibodies. Well, the other thing that we just talked about was Rh. But Rh, there are no bacteria in the gut that have Rh. There just aren't any. And so, unless you've been exposed to Rh blood, you're not going to have any anti-Rh in your bloodstream. You just don't have it. The only way you're going to get it is if you have exposure. Well, that becomes important when we have an Rh positive male and an Rh negative female. Because what's going to happen is their children can be either Rh positive or Rh negative. Well, if they're Rh negative, there's no issue, no problem. No problem whatsoever. Because in other words, when this little child was in the mother's fetus, in the mother's uterus, both the baby and the mother had the same Rh status. So there's no problem. Well, what happens is sometimes when you have an Rh positive father and Rh negative mother, the offspring are Rh positive. And so now in the mother's uterus, there is an Rh positive baby, which is the opposite of the mother. So it looks like this. So there's the father, he's Rh positive, there's the mother, is Rh negative, and there's the baby, it's Rh positive. Well, this baby usually doesn't have a problem because there's no mixing of blood between these two except at birth. So at birth, there's a lot of bleeding that happens. And the placenta, which has both the mother and the fetus's blood in it, gets ripped off of the wall of the uterus, and there's a lot of bleeding that happens. And when that bleeding happens, the blood of the fetus mixes with the blood of the mother. It gets in her bloodstream. Well, think about it. In her bloodstream, she's Rh negative. But here comes this Rh positive blood. Let's go back to this, what we talked about before. If she's negative, what is Rh to her? Is it self or non-self? To this person here, is Rh self or non-self? It's non-self. And remember what happens when non-self gets in your blood or gets exposed? We're going to make anti-Rh antibodies. So now floating around in her blood is anti-Rh antibodies. Well, it's not going to hurt this baby because this baby's gone. It's been delivered. It's a kid now. It's fine. But if she ever has another baby, another fetus, and it's Rh positive also, it's not like it was before. Look over here, there's no antibodies. But now, because she's been exposed to the blood, she has antibodies. And those antibodies can cross the placenta. And what they'll do is they'll go to that fetus's blood and they'll destroy the blood in the fetus. Well, think about what happens if you have all of your blood destroyed. What do you think is going to happen to the fetus? What's going to happen to this fetus? It's going to die, exactly. Either before birth or right around birth or after birth, but it's going to die because it's not going to have any red blood cells. And you get a condition which is called either Rh disease, but technically it's called erythroblastosis fetalis. Because what happens is the red blood cells get destroyed and the fetus dies. Does it make sense? So again, she's negative. The only way it's ever going to have a problem is if she's negative and the fetus is positive. That's the only way it's ever going to have a problem. And when that fetus's blood mixes with hers, usually that happens at birth, she's going to make these anti-Rh antibodies. And they're going to float around. And then again, if she ever gets pregnant again with another Rh positive baby, those antibodies are going to enter, they're going to go across the placenta, and they're going to destroy that fetus's red blood cells. So there's no problem with her first baby. So the first baby is going to be healthy. But the second baby is probably going to die. And the third baby is going to die. And the fourth baby is going to die if they're Rh positive. So before we knew about this, a man and a woman would get married and they would have one child normally, and then every time that they got pregnant after that, they would have a miscarriage. So if you go to really old cemeteries and you look at them, so there's the man and the woman, their headstones, and then you're going to have like 7 or 8 or 3 or 4 or whatever little headstones for their babies where they were either stillborn or miscarried. So the way you fix that, and it seems kind of weird, but you inject the mother with anti-Rh antibodies. It's called RhoGAM. GAM stands for the fact that it's gamma globulins. And that RHO stands for RH. And what it does is when you inject her with anti-Rh antibodies, when the baby's blood mixes, it very, very quickly gets rid of that baby's blood. It clears it before the mother reacts. It has to be done with every birth. Any questions? Anybody else? Any questions? Can everybody figure out a blood type? So if you were given pictures like this, would you be able to figure out what this person's blood type is? What this person, and so on. Everybody do that? Can everybody figure out who can give to whom? So here's that chart, but you need to make sure you can understand who can give to whom. Any questions about it? Does everybody understand why type O negative is the universal donor and why type AB positive is the universal acceptor? So Mahala says, what's going to happen if the baby's negative and the mother's positive? Nothing happens, actually. Because if the mother's positive, think about it. Here's the mother, and she is Rh positive. So for her, what is self? I mean, what is Rh? Is it self or non-self? It's self, right? So is she going to make any anti-Rh antibodies? In other words, she'd be killing her own cells, so she's not going to have any, right? So that's not a problem. But then also think about the baby. The baby is Rh negative. So on the red cells, is there any Rh on there at all? So even if there were anti-Rh antibodies, there's nothing to attack. And so that is never a problem. It's only a problem, only in one situation. And that is where the mother is Rh negative and the fetus is Rh positive. So if you're a female and you go to get pregnant and you go to your obstetrician, if they take your blood and they're going to check, if they see that you're negative, they're not even going to ask you who the father is. They're just automatically going to inject you with this RhoGAM. And it's usually 2 or 3 injections. And that happens just before and right after birth. And again, This is anti-RH antibodies. That's all it is. Just like when we did a blood test, we did it in a rabbit, remember? And we took his blood. That's what it is. It's just the same thing that we used when we check out and determine blood types. It's the same thing right there, so that we use to check these blood types. Any questions? Okay, so that's the end of that material, and that's basically as far as we're going to go today. So I'm going to go ahead and stop the recording, but don't leave yet. I want to ask you a couple of questions.